Okay. So biology is a cumulative course, which means that everything that we've learned in the first 2 units of the class, as well as unit 0, still come into play in this 3rd unit, and we will continue to build upon the foundational knowledge that we've gained from each successive unit. So Sternger was a concept that we actually introduced on day 4 of the school year, way back in unit 0, and it is still something that is important as we start unit 3. So we can't forget about Sternger. We can't forget about those 8 essential life processes that organisms must be able to work towards and achieve in order to survive. And today and through this 3rd unit, we're going to focus specifically on one of the R's, and that R is reproduction. Reproduction. Additionally, tomorrow starts the beginning of National Hispanic Heritage Month. And in recognition of that, as well as in order to make the slides a bit more linguistically inclusive, I'm going to be including Spanish translations for most of the words that you'll see on the slides today and going forward. So you don't obviously need to write down both languages. Whatever your preferred language is between English and Spanish, that's the one you should pay attention to and start getting in the habit of taking notes. Additionally, you'll see that I've highlighted the words that I want you all to write down in yellow if they're in English, and then in green if they're in Spanish. Again, you don't need to write down both, but I want you all to get in the habit of taking those efficient notes. And so I don't want you to write down everything. And in fact, there will be some slides where there's nothing for you to write down. You just need to listen to me talk. But when you see something that is highlighted, that is a clue that you should write it down. So please make sure that you have your notebooks out and you're ready to not only refer back to your previous notes, but also begin taking some new ones as well. So our 3rd unit is on cell reproduction. And reproduction is the production of offspring by a sexual or asexual process. So we're thinking about offspring. We're thinking about how new cells and new organisms can be born out of older cells and older organisms. So reproduction is used to make new cells, but it's also important that we note that reproduction is used to copy and preserve DNA. Okay, so there's nothing on this slide that I really need you to write down because we're going to break this down into far greater detail today. But just keep in mind that most cells will reproduce in their lifetime. Not all cells, but most cells will reproduce in their lifetime. Now, here's where I want you to start taking your notes. As you can see, I've highlighted things that I think you should write down. So cell division is the process by which individual cells and unicellular organisms reproduce. I'll give you some time to write that down while I go grab my water bottle. Okay, so hopefully that was plenty of time to write this down. But this is what we're thinking about, cell division. Now, cell division is essentially the same thing as cell reproduction. So you'll hear me use the two.
Can you go back real quick? All right.
Yes. So cell division and cell reproduction are essentially the same thing. So you'll hear me use the two terms interchangeably. And what we see here is that cell division actually taking place. On the left, we've got a plant cell. You can see that the orange material is actually chromosomes, and they're being pulled apart. They're being split apart. And we're starting off with a number of chromosomes inside of the cell, inside of a single cell, and they get pulled apart. And then additionally, those two cells then get pulled apart as well. So what starts off as one cell is then pulled into 2. On the right, we can see the exponential growth of a colony of bacteria. So exponential meaning that 1 turns into 2, which turns into 4, which turns into 8, then 16, then 32, 64, 128, 256, and so on and so forth. All right, so this is not just going 1, 2, 3, 4, 5. It's growing exponentially, right? So very, very quickly, you end up with a very large colony of bacteria. So I want you all to think about it before I talk about it, and we're going to go into depth in the next few slides. But before I talk about it, why do we think new cells are created? What's the point? Why are new cells created? I'm waiting.
I think new cells are created because I really don't know, like, because I don't want to be wrong.
No, there's nothing wrong with being wrong, and I don't think you will be wrong. I have confidence in you, Michelle.
I think new cells are created because our body go through changes.
Excellent. Yeah. So our bodies change. You're not the same person you were when you were first born. So you have to grow. That's one of the reasons that we make new cells. And our bodies are constantly changing. So that's a great point. Thank you, Michelle. Anybody else have any other reasons? Okay. So there are 3 primary reasons why cells are created. But before we get into those reasons, I want to do a quick flashback and gauge what you remember from the previous units. So my question is, which of the 4 macromolecules is DNA? And you can feel free to type into the chat if you're too nervous to come off of mute. Which of the 4 macromolecules is DNA?
I really don't know, but can you name the 4 macro?
Okay, good, thank you. So we've got 4 different types of macromolecules. So hopefully you all, thank you, Nick, that is the correct answer. Hopefully you all have your notes and you can refer back to them. But we've got 4 different types of macromolecules. I'm typing them into the chat now. Carbohydrates, lipids, nucleic acids, and proteins. And Nick is correct that DNA is a nucleic acid. So we cannot forget these concepts, people, because they're not going away. We will continue to be tested on them, and they're still important as we master the material and master these concepts. So you've got to be willing to go back into your notes and have access to things that we've talked about 3, 4, 5, 6 weeks ago. Okay. So I'll move on, but that's just a good review. There's nothing that I really want you to write down on this slide, but this is further thoughts about DNA. We know that DNA is the genetic material. So when we think about genes, we should be thinking DNA. And then there are also these other terms that we're going to introduce in this unit that should also conjure up thoughts about DNA. They should also make us think about DNA. They are chromatin, sister chromatids, chromosomes, homologous chromosomes, and genes. Okay, so for me, even after having taken 3-4 biology classes, it is still difficult for me to fully understand some of these differences between these terms, primarily because they sound so similar. We've got chromatin, chromatid, chromosome, chromosomes. So we've got to keep in mind that all of these things are referring to DNA and genetic materials in some way. But there are some important differences between them. So again, you can see that there are words that are highlighted in yellow, English words that are highlighted in yellow, and there are Spanish words that are highlighted in green. So this is what I want you all to write down. Okay. So chromatin, if we're looking all the way at the bottom, it says chromatin is random and disorganized DNA. Chromatin is random and disorganized DNA. So in that image on the left, we can see a picture of chromatin. We can see chromatin inside of a nucleus. Chromatin essentially looks like a bowl of spaghetti, a whole bunch of noodles just tangled up. There's no real order to it. And there's a reason why there's no order, but we won't talk about that right now. But one of my students in my previous class asked me that. So if you're wondering why does it look like that, there is a reason. But it's just random and disorganized DNA. There's no order to it. But chromosomes, on the other hand, are condensed, meaning they're ordered, and they're organized. They're tightly wrapped around a specific structure. So that's the primary difference between chromatin and chromosomes. Okay. So that's the difference between chromatin and chromosomes. And what I would do here, as I would, if I were you taking notes, I would draw an X on in my paper, just like we see here. We've got basically an X, and then I would begin to highlight different parts of that X. So the ends of the X, the top and the bottom of the X, are called telomeres. They are the ends of a chromosome, and they protect the chromosome from deterioration. So they protect the chromosome from breaking down. And they also protect it from fusion, meaning we don't want that chromosome to fuse or to connect with another chromosome. We want it to remain separate. So those telomeres protect the chromosome. Each chromosome is made up of 2 sister chromatids, and they are identical copies of the DNA. So one half of the X is one chromatid. The other half of the X is another chromatid. We call them sister chromatids because they are identical copies of the DNA. And then this region in the middle of the chromosome, where the 2 sister chromatids meet, is called a centromere. All right. So again, you don't need to write down all of the notes. You just need to write down the notes that are highlighted in yellow and those that are in your preferred language. All right. So we should be familiar at this point with the cell. We dedicated our second unit of the course to the cell. And so we know that eukaryotic cells have a nucleus. But we can zoom in further. We can zoom in not only into the nucleus, but further than that. But we know that the cell's genetic material is stored in the nucleus. So that's what you should write down here. The cell's genetic material is stored in the nucleus. So that's step one. Then we zoom in further to step two. Within the nucleus, the genetic material is organized in chromosomes. And then lastly, chromosomes are tightly wound DNA. So the DNA is really tightly wrapped up to form a chromosome.
All right, Mr. Wright. I'm gonna need you to go back to the first step. I'm gonna take a picture because you move way too fast. I write too slow. All right, you can go to the second one now. All right, you can move it.
Again, you only need to write down what is highlighted in yellow. So chromosomes are tightly wound DNA. Humans have 46, meaning we have 23 pairs of chromosomes. We get 23 from our father, our biological father, and we get 23 from our biological mother. So in total, we have 46. Other organisms have more or less chromosomes, and the number of chromosomes that you have does not necessarily correspond to the level of sophistication of the organism. So, for example, humans have 46, but fruit flies, which are in most ways less sophisticated organisms, have about 98 chromosomes. You don't need to write anything on this slide. This is just a picture and I can talk about it. So our cells' bodies are constantly dividing, and keep in mind that dividing and reproducing are used interchangeably here. So most of the cells in our body are constantly dividing. Not all, but 95% of them. And they have to do this for specific reasons. We'll talk about that in a couple slides. But they do this through a process called the cell cycle. And in general, there are two cell types. There are gametes, which are sex cells, and then there are also somatic cells. Again, we should really only be writing down what's highlighted. The gametes, which are also called sex cells, are sperm and eggs. So these are the cells that um, allow humans and other multicellular organisms to reproduce. There are also somatic cells, which are essentially everything else besides the sperm and the egg. So the prefix soma, S-O-M-A, means body. So we can think about somatic cells as being body cells, everything else besides sperm and egg, all the other cells in the body. So this difference in the type of cells is important because these cells, and we'll talk about this in greater depth in the next couple of days, but these different cells have different numbers of chromosomes. So your somatic cells have all 46 chromosomes, but your gametes, the sex cells, have half of that. They only have 23. Okay. So why do we need to make new cells? Again, we're only writing down what's highlighted here. So this should be just one, you should write down the question, why are new cells made? And then you should write down one, replacement. So we have to replace old cells as they die. And this is happening constantly. In fact, cells have a programmed death. They are within their DNA, they are told exactly when to die. And so once that happens, they need to be replaced. And that's one of the primary reasons why we make new cells. We also make new cells for the purposes of growth. And we know that most organisms grow in size over time. So from birth to toddler to pre-adolescent to adolescent to teenager to young adult, so on and so forth until you're elderly, there are constantly changes taking place in our bodies. And there's certainly some amount of growth, but once you reach a certain age, you're not actually growing. You're not getting taller. You might get bigger, but you're not getting taller. But there are still important changes happening in your body. Some organisms get bigger. I'm sorry, some organs get bigger within our bodies, and some organs actually get smaller within our bodies as we get older. Some organs become better at their jobs as we get older, and some organs get a little bit worse. So there are these changes, and we have to make new cells to account for those cells. And lastly, we make new cells for the purpose of reproduction. And reproduction can be both sexual and asexual. The goal is to produce new offspring. We need new cells to do that. Okay. So this video really illustrates the division of cells within a plant cell. The orange stuff is the genetic material, so we see it being pulled apart. We can see new cells forming as the genetic material is pulled apart. All right, so again, we're focused on what is highlighted. That's what I want you all to write down. So the cell cycle is the process that cells go through to make new cells. And there are different stages or different phases of the cell cycle. And we're going to talk about these phases in depth. But essentially they are interphase, which is itself made up of different stages. Then we've got M phase, M as in matter, M phase. And then we've got cytokinesis after M phase, and that's the last step of the cell cycle. So again, we'll talk about each of these steps in depth, so you don't need to write that down. Just write down the definition of the cell cycle. It's the process cells go through to make new cells. The process cells go through to make new cells. Okay. And again, here are the three major parts of the cell cycle. So this is where you should write this down. The three major parts of the cell cycle are interphase, M phase, and cytokinesis. Interphase, M phase, and cytokinesis. Okay. So interphase is the first phase of the cell cycle, but it's also the longest phase.
Can you go back real quick? I can go.
So only the things that are highlighted on this slide need to be written down. So interphase is the longest phase of the cell cycle as the cell prepares itself to divide. And interphase has 3 steps. There's G1 as the cell is growing. There's S, which stands for synthesis. And synthesis is very important, and I'm going to come back to that. And then there's G2, which also stands for gap2, and the cell is continuing to grow. So in both G steps, G1 and G2, the cell is growing. During synthesis, the DNA is replicating. And we say it's synthesis because new DNA is being made. There is a copy of the DNA being made. And we're going to see this in an image on the next slide. So interphase is the longest phase, and this is the preparation for division. The cell is growing, and the DNA is being replicated. All right. So DNA replication kind of looks like this. And again, you should be writing down the words that are highlighted. During replication, the DNA is copied. Essentially, the 2 strands of DNA, so we see the double helix here, they get unwound, like they get unzipped. And so now we have access to each strand of the DNA. And then each strand gets copied. So there's a new strand added to it that is an exact copy of its old partner strand. And then eventually, as we continue to unzip the DNA further, as we continue to unzip the DNA further and further up, we continue to make a newer and newer copy of the DNA until eventually we're left with 2 new DNA. Okay. So during replication, which is during synthesis of interphase, the DNA is copied. That's what replication means here. The DNA is copied. Interphase, we move into M phase. And there are 2 potential M's. There are 2 types of cell division. There's mitosis, which is asexual reproduction, and there is meiosis, which is sexual reproduction. We will talk about mitosis and meiosis in greater depth over the next 2 days, but I do want you all to write this down, which you see it highlighted on this page. So mitosis is the type of cellular reproduction that somatic cells undergo. So all of the cells that make up your heart and your eyes and your skeleton, your muscles, these undergo mitosis. But the cells that make up sperm and eggs undergo meiosis. And those, of course, are called your sex cells, aka your gametes. Okay. So this is a good image representing what mitosis looks like. Start off with one cell. Eventually that one cell begins to split. And eventually you end up with two distinct cells that have their own membrane, their own plasma membrane. And then lastly, after M phase, we have cytokinesis. Cytokinesis literally means cell movement. Cyto means cell, kinesis means movement. The cytoplasm and the cell membrane of the parent cell split into two cells, and we call those two cells the daughter cells. So cyto means cell, kinesis means movement. We put the two together. We're talking about how the cells literally move apart from one another. And what starts off as one parent cell ends up being split into two daughter cells. So we can see that happening a little bit here. We can literally see the cells moving away from each other. You can't really see how they start off as one cell. What's that?
Can you go back, please?
Okay. Hopefully that's enough time.
Yeah, you go ahead. He said that.
So you can't really see in this animation how they start off as one cell, but essentially they get cleaved, which means they get separated from one another. And then cytokinesis allows them to move apart from one another. They're moving apart from one another. So we put it all together. If you haven't written anything down up until this point, this is your bang for your buck slide. You want to take a picture of this. You want to write this down. But here are the steps. We've got interphase, which of course can be separated. It's the longest phase, so we separate it into G1, synthesis, and G2. G1 is a period of growth. Synthesis is the period when the DNA is replicated, and G2 is another period of growth. All right, so those are the three steps of interphase, G1, S, G2. Oops. Then we have M phase. M phase is mitosis or meiosis. And lastly, we've got cytokinesis, which is when the cells move apart. All right. So all you need to write down on this slide is the definition of cancer. It is an unregulated growth of abnormal cells. Unregulated growth of abnormal cells. Oftentimes, if left unchecked, this can lead to a tumor. In this case, we're seeing a tumor in someone's colon. Does anyone have any thoughts as to why this unregulated cell growth occurs? What causes this unregulated cell growth?
Mutations.
Okay, yeah, mutations are a big reason why. And so each cell, of course, has DNA. Each cell has an entire copy of the DNA, but each cell has its own unique DNA. And so sometimes some of the cells carry mutated versions of the DNA. And sometimes those mutations can lead to cancer. Good job. Good answer. All right. So our cells are very smart. And exactly what Brian just mentioned, the mutations can sometimes mess up the intelligence of the cell. So normally cells have checkpoints that regulate the cell cycle to prevent mistakes by stopping growth and division if necessary. So along the cell cycle, there will be these checkpoints to say, uh-oh, if something's wrong at this point, then we're going to stop. Don't divide. Don't grow any further. But if you're good, okay, now you can move on into synthesis. If something's wrong here at synthesis, well, we're going to stop. You can't divide. You can't grow any further. But if everything's good at this point, then we move on to G2. If something is wrong here, we've got another checkpoint. If we're not ready to divide then we say, okay, you need to stop. Don't grow any further. And then lastly, once we actually undergo M phase, once we undergo mitosis or meiosis, if something is wrong there, well then the cell is not allowed to split. All right, so there are these checkpoints that are intentionally designed within the DNA of the cell to say like, okay, we're good to go, or no, we need to hold up, something's not right. And if those checkpoints aren't there, or if they fail to work, then we can end up with cancer. Question? Okay. So again, here's some of the questions that the cell might ask itself. Of course, this is all in the DNA as it moves through the checkpoint. So at G1, is my environment okay? So is it too acidic or is it too basic here, or am I good to go? Is my DNA intact? If my DNA is damaged, then I don't want to copy it because it's already messed up. And we get over here to the G2 checkpoint. Has the DNA been correctly replicated? Before I start mitosis, have I replicated my DNA? Then I get to mitosis or meiosis, and then I want to ask myself, are my chromosomes ready for separation? to actually become 2 new cells. So those are some example questions of the checkpoint. All right, that's it for the lesson for the day. We've got 18 minutes, so I apologize that the synchronous portion ran a little bit long, but you only have one assignment and you have an exit ticket. And I think you have plenty of time to knock them out. The exit ticket is 5 questions, and one of them is just asking you how you are doing today. And the cell cycle practice is, I believe, 8 questions, and so it shouldn't take long either. So go ahead and knock those out, and you'll be in a good place following our first day. I'll be here if you have any questions or any issues accessing them. Again, I'll show our Canvas. So what you should be focused on is the practice quiz and the exit ticket, which is right there in the normal module, the Unit 3 module. Oops. So this is what we're focused on right here, those 2 assignments. Yes, thank you for reminding me. I know some of you still have to take that test from last week. I'm not sure if SchoolNet is working, so I'm gonna put it on my plan is to use Canvas to do it. So tomorrow, make sure you ask me again, Joseph, but thank you for reminding me.
This D go hurt, bro.
Was that test important what we took last week?
Was it important? Yes, it is important.
Like, is it gonna is that the final grade? No, never mind.
It's not I mean, it's it's just we'll take a unit test at the end of every unit. We've got 10 units, so each one is important, but it's not it's not going to make or break you. Okay, folks, it is 1:20, so I will stop sharing and I will stop recording. If you need anything between now and tomorrow, please let me know. Please do make sure you're